So, students, it's already 4:30. So most of you have joined already. It's my pleasure to see you all. You are most welcome to the second day of our workshop, and I hope you are all awake, quite excited to go to the next chapters of biochemistry um, that we will be dealing today. So let's at least 30 members to join in, then we start. And how are you, everyone? Staying at home? In Delhi, it's horrible. The cases are increasing like hell. So even for getting a packet of peanuts, we are thinking 100 times, shall we get it or not? Um, in Delhi, it's horrible actually. Even India, it's become the second largest state, second largest country to have the COVID. And soon, I mean, it's four something, four was the figure, 4 million or trillion, 4 million maybe. And it's soon reaching to the USA one with the 6 million. So that's quite a horrible thing to think. But anyhow, just even when I was teaching for my MBPT students, some of my students got COVID positive, but they are all fine now. Um, it's just take care of yourself. Somebody is coughing outside, just create a distance. Never ever forget to wear your mask because this really is a good protection. Really, it is because one of my friends in USA, they are they are like very at higher position of research scientist in Boston, and they are telling to the chief ministers of Punjab about the various details how you should be protected. So whatever the news channels and everyone is saying to us that keep washing your hands and covering your face with mask, these two regimes has to be maintained properly, then you could be protected from the person who is infected, because most likely everyone is affected at this moment, and we never know uh, yeah, when we will get affected. And it's all depend upon our immunity of the body, how strong is our immunity. If we have a strong immunity, then things will be fine. Otherwise it take some time to get recovered out of it also. So no worries, chill out. Uh, we will remember these days, and we will tell to our grandchildren if we are alive during this time, and how we survive COVID time, with funny stories actually. Anyhow, I talked a lot. Um, I want to start with you two guys today. Um, with the enzymes. And yeah, any questions from yesterday? Any things that you have anything in your mind at the moment before we start? Good evening, everyone. Good evening. Just a second. I'm getting my charger to be done there. So students, also from the yesterday, if you the recording was not done good, but if you want to see the recordings of our lectures, I tried to record on the YouTube, but it
Hi,
was
today I have an easy question for you. I hope that most of you would be able to solve it right away, but for those of you who cannot solve it,
sorry. Please subscribe to my channel also if you are not aware. It's Simranjit Singh. So I will share the link of yesterday's workshop here with you. So somebody or something went off and they were not able to join us. So you can join me via this link if you want recorded version of this. So channel looks like this, Simranjit Singh. So please subscribe, share, and this will give me motivation to create more videos. So I have already started with the series of uh, NEET exam for the class 12th, and also will try to start series for the GATE exam and NET exam, CSIR NET exam. So I just need your motivation. So I just need some ideas because I'm all alone in this world of YouTube, but I have a lot of content to teach, a lot of things to explore with you students. So just need your guidance also while going through the path because you are our sole path for that. So I will not talk much. So I think we should start now. Uh, so let's start with the enzymes. This one or that one? I think that one first. So all we ready, everyone? Everyone is set up there to start the class? Oh, sorry, I haven't shared my screen. Oh, thank you so much. Sometimes these small glitches happens. I hope you can see the video now. Yeah, I'll maybe try this one. Everything is fine, I guess. Can you see the videos? Now I can share my screen with you. I think things are fine. Please thumbs up if everything is clear. Beyond time. Okay, it's visible now. That's good. Mhm. Awesome, awesome. Okay. Um, so I had shared with you the objectives today. Please join in. Uh-huh. So I already had shared this biochemistry objectives that somebody was asking me yesterday. So we are today dealing with the enzyme part of everything of its, from its history, nomenclature, classification, kinetics, its inhibitors, their importance, um, regulations, isoenzymes, everything we will be dealing with it. And beyond that, amino acids we covered yesterday quite well. Nucleic acid we will cover about structure of DNA in subjective manner with a central dogma of. I showed you some videos, but I want to also have you some subjective knowledge about it because you can't tell in your exam or anywhere your videos there. So lipids we are done also. So some of the things that are left, and then one of your two virtual labs are left. That is PCR and gel electrophoresis. So first you isolate your DNA, then you increase your concentration of DNA by a polymerase chain reaction. Then after polymerase chain reaction to check how much DNA you have extracted with a short or medium or long chains, you do it with the gel electrophoresis. Yeah. So in this way, you can arrange your we will arrange over this workshop. And also we will deal with something with disturbance in the thyroid function, liver function test, and jaundice also. These three is we will be dealing today also. So let's start to the second day of our workshop. Uh, there you go. 7th. So anybody join, somebody ask in the WhatsApp group. Everyone is joined, right? So everyone is good there. Oopsie. So please do not disturb on that WhatsApp. Okay, let me start. Concentrate, focus. So, um, so somebody has to unmute themselves. Somebody just got unmuted, please. So enzymes and mechanism of actions. So basically we will be dealing with the basic part of how enzymes works, what is the mechanism, and what are the various principles that made them to work like very specific to the things. We will discuss about lock and key methods. So all these things we will be dealing in it. So let's start. So and basically you will be familiarized with the with the biocatalyst, what a biocatalysts are, and to study their types of the structures. Beyond that, to study the mechanism of action, what is the specificity of enzymes and its functions. So that's five parts we will be dealing in the objectives of our enzymes today. So, what is enzyme? Anyone can tell me what is enzyme is? Still people are joining. What is enzyme? Just type in. Anything that you know about enzyme before we start. What is your knowledge of what enzyme is? Hello? Hello students, you still available? Ah, okay, it was not working, sorry. So voice is fine? Biological protein catalyst, okay. So I didn't made this up, so sorry for that. Yes, that act as a biocatalyst. Great. Proteins are all enzymes are proteins. That's one of the nice statements Sneha just tell us that all enzymes are proteins, but all proteins are not enzymes. So biocatalysts that speed the metabolic process. True, true, true. So nice statements. That's what the basically enzyme is. Oh yes, Nitish Kumar, you made a very nice point that they increase the activation energy of a chemical reaction actually. So that's why they are so important for us. Just give me 1 minute. My charger is my laptop battery is not getting charged. If it is not working, so we might be in some trouble then. I hope it will work now. Work. I need to fix this. Maybe over the weekends I do this on my own. Eureka! We fixed it. So we are good to study about enzymes now. So let's start. I'm very excited. I hope you all are. Um, so enzymes are the biological catalyst which increase the rate of reaction by lowering the energy of activation. So whatever the energy that requires to run the reactions, we lower it down and then we increase the rate of reaction by increasing its rate of reactions. That's what the enzymes are doing. So they catalyze the nearly all chemical reactions taking place in the cell of the body, not altered or consumed during reactions, and they are quite reusable also. Yeah. Important terms to understand biochemical nature and activity. So they have one active site, these enzymes have, where the substance of substrate attached to it, and it is called active state site. So enzymes are usually very large in proteins, and active site is just a small region of that enzyme molecule, very small region attached to it. So this is how the active sites looks like. So enzyme molecules contain special pockets or a cleft called active sites. So here you can see a substrate entering the active site of enzyme. This is your enzyme it is, and this is your active site. Uh, so some people wants to, they are late. Oops, but just unmute yourself. Oh no, it's so much noise coming up. So So they are the like so you can see here there's the active site and your substrate has to be attached there on top, and an enzyme could change the shape or slightly as a substrate there. So they come and attach to it. They make an enzyme substrate complex. So this complex later on being changed into the enzyme product complex EP complex here, and then they are moved as a product later on. So this you have learned zillion of times in your plus one plus two studies, but we will go through them once because the basics are quite needed. Things got later very complicated. um, Substrates, the reactant in the biochemical reaction is termed as substrate here. When a substrate is combined with enzyme, it forms an enzyme substrate complex. So you have your substrate which joins to the enzyme. So enzymatic reaction substance, the substance at the beginning of process on which enzyme begins, its action is called as substrate. First is substrate, then it joins it, then it's become product, then it leaves the enzyme. So this is an active side where it is joining it himself. Our substrate. Then this is your lock and key model. So in the lock and key model of enzyme action, active site has a rigid shape. So it has a rigid shape with only substrates with a matching shape can fit into that. And substrate is a key that fits the lock of the active site. This explains enzyme specificity, and this explains the loss of activity when enzymes denature. So you can see this lock has specific shape and the substrate which is acting as enzyme, and this is a key which is acting as substrate. So there's a correct fit and will react to that. So you will see some results out of it. But on the other side, uh, here at the bottom, if there is no extra a key to that, this extra substance to it, the key, so this is incorrect substrate, and it there you will see no reactions to it. So this is a very common lock and key method. Then comes your apoenzymes and holoenzymes. So apoenzymes are protein portions which are inactive, and enzyme without its known protein moiety, which does not have any moiety to it, they are known to be as apoenzyme. Students, come on time. This is so we cannot see the slides. Really? Everyone, can you see the slides? So I'm getting messages that I cannot see the slides. So I am presenting. So if I stop presenting, then you can't see the slides. So most of them you can see it then. And the voice is also fine, right? So please check your systems, bache, student, if all is fine with it. Everyone, Most of the students, it's fine. Please check your system. Sorry for that. Uh, so the apoenzyme is the enzyme without its non-protein moiety is termed as an apoenzyme, and it is an inactive one. And whereas the holoenzyme is the one with the active enzyme in it with its non-protein component. So you can see here is the apoenzyme with a no coenzyme attached to it. So it has now cofactor, which is a non-protein portion, which act like an activator. So this will attach to the apoenzyme, will make it into the holoenzyme, and then after turning into active form, it can attach to substrate for further functions. So what the term came cofactor. So cofactor is a non-protein chemical that is bound to an enzyme and is required for the catalysis here. So there are types of cofactors, that is your coenzyme and prosthetic group. So what are coenzymes? The coenzymes are the non-protein component loosely bound to the apoenzyme by non-covalent bond. So they are attached to the non-protein component with the apoenzyme by non-covalent bond. Examples are vitamins or compound derived from vitamins. So they all act as a coenzyme. Whereas a prosthetic group, they are tightly bound by covalent bond by the apoenzyme, and that is called as your prosthetic group. So that's the main difference between your coenzyme and prosthetic group. And how is the enzyme specificity? It is there. So enzymes have varying degrees of specificity for substrate. So enzymes may recognize and catalyze a single substrate, a group of similar substrate, or particular type of bond. So you can attach to any part of it. Uh, Enzymes can attach to any part, can recognize and catalyze further. So it has varying degree of substrate specificity for substrate. So all chemical require some amount to energy to get them started. The first push to the start the reaction is called activation energy. So here you can see a lactose molecule is there. To break it, so you need some activation energy without enzyme. This is the activation energy required without enzyme. So after the it is reached to the point where it is broke, then this is the decrease in the net energy released from the splitting of the lactose. So at the end you have glucose and galactose after splitting your lactose. So you have your decrease in the activation energy like this. So instead, so you can see that the activation energy is quite high to break these two bonds. So here there is no enzyme, so it was done just by their own. So that's energy to start the first reaction. This whole energy is called your activation energy. So just summary of what we studied so far. So enzyme has a complex or hollow enzyme, or it could be simple only protein. If it is a complex or hollow enzyme, so apoenzyme is required and cofactors are required. So apoenzyme and cofactors will have prosthetic groups and coenzyme. So coenzyme will include large organic molecules loosely bound to apoenzyme, whereas the prosthetic groups includes usually small inorganic molecules or atoms usually tightly bound to apoenzymes. So that's how the enzymes and holoenzymes, cofactors, apoenzymes, they are quite correlated and interconnected to each other. Mhm. So next part, mechanism of action of enzyme. So how does they work? So they increase the reaction rate. So what we saw before last time, so there was activation energy, which was quite high, but they decrease that activation energy by increasing the reaction rate. So that's what the enzyme are working way. So enzyme substrate interaction. So formation of enzyme substrate complex by lock and key model or by induced fit model. So, so when a substrate fits properly in an active site, an enzyme substrate complex is formed. Like this is called ES complex. So E plus S give rise to ES. And within the active site of ES complex, the reactions occurs to convert substrate to product. So ES complex they comes together and they give rise to a product. So like ES, this substrate together give rise to your enzyme plus product like that, and the product are then released allowing another substrate molecule to bind enzyme. So your substrate is molecule is free. Now it's available to add to another, and it's reusable, and this cycle can be repeated millions of times per minute. So this could be run million of times per minute in your reactions. So the overall reaction for the conversion of substrate to product can be written as, so E plus S, which is a reversible reaction, giving rise to ES complex, and this ES complex is giving you energy plus, sorry, enzyme plus your product. So students, be on time. You are coming quite late. So where is the link for this now? Maybe I join them here. Okay. Uh-huh. So that was something about basics of enzyme substrate complex, how they are working out. So in a very simple way, in a very nutshell way, it has been explained their action. This is with the animation now. In the first step, we have seen E plus S giving rise to ES. So like that. So you have a complex, and then enzyme product complex is formed with this ES with the EP transition state and EP. Then you have your enzyme product. There's a transition state in between from your enzyme substrate complex to enzyme product complex. So end product is your enzyme plus product, like this. The product is made, so enzyme is ready for another substrate. So enzyme is again reusable for million times to go for further reactions. So here you can see this study provides a complete detail of enzymes, and it introduced enzyme as a biocatalyst and explained the mechanism of enzyme reactions. So it explained everything to us now, about that you have understood now at least, what is enzyme working as a biocatalyst, and how is the concept of mechanism of enzyme action works, how this study complete detail of your enzyme is. So these are the references. So thank you very much. So that was a little bit some introduction about enzyme and mechanism, just to give you a hint how things are working out in the case of mm, an enzyme world. Somebody please unmute themselves. So your sound is coming to us. Oh, some fan sound maybe. Deep Narayan, could you just unmute yourself, please? Just check. So let's start with the second session of today. Uh, Sorry. Hello. Yes. Okay. All good. So let's come to the nomenclature of enzyme and its various other parts that we have to discuss with it. How, what are the various types of enzymes are and how they are working. Something about allosteric enzymes, something about inhibitor enzymes, and yeah, what are their application? What are their yeah, it's various different things that you will undergo through this part. So this you can see you will acquaint with the nomenclature and classification of enzymes. You will understand the specificity of enzymes. You are able to understand the allosteric regulation, and also a little bit about feedback mechanism. So these four topics will be covered in this. So you can see how enzyme and substrate, they are coming together at the active site and letting in and letting it work to be product later on, and working in a yeah, in its functioning manner. So, so in this content, we are dealing here in the first part with the nomenclature and classification of enzymes, ES complex, a little bit about cofactors and coenzymes and proenzymes. So this is what we'll be dealing. So first of all, in the nomenclature and classifications, enzymes are classified by placing them in a categories according to the reactions that they are catalyzed. So they are oxireductase, transferase, hydrolase, lyase, isomerase, ligase. So you can see in each end we are adding A's to the enzymes, and initially it's like their functions, what their functions they are doing. We have their name like oxireductase, something to deal with the redox reactions, transferase, transferring one enzyme, hydrolase, removing some, breaking some bond with the help of water, lyase, some joining it, then isomerase, changing the shape of your giving isomer of your structure, ligase, joining two structures. So as the name is, so the enzymes are working. So oxireductase catalyze your redox reactions. So you can see here reductases and oxireductases. So here is your lactate and this is your pyruvate. So our lactate with the NAD plus gave rise to lactate dehydrogenase, yeah, lactate dehydrogenase, and giving rise to pyruvate and an NADH. So here lactate dehydrogenase is changing the structure of your COH with a hydroxide bond to the C double O bond. So this is how it's becoming more into the other form, from the oxidase to the reduced state. Also, some transferases are also known from transferring of the one molecule to another. So like transminases is a catalyzed transfer amino group of amino groups, and kinases transfer the group of phosphate groups. Same way here, there's an example of PNMT. What is it doing? This enzyme is it is removing the methyl group donor. So it's attaching the methyl group donor to be attached to this amine group on that side, like this CH3. So PNMT being attached this methyl group like on the other end. Then classification of enzymes. Hydrolases cleave bonds by adding water. Uh, So phosphatases, peptidases, lipases, so these are all added to the, they are just cleaving the bonds by hydrolysis by adding water. So phosphatases, peptidases, lipases are the example. So here you can see there's a triglyceride molecule, and with the 3 molecule of H2 and a lipase, so we are having a glycerol and 3 molecule of fatty acids. So we are destroying that, breaking up that bond of glycerol by via water with the help of enzyme lipase here. And same way, lysases catalyze the removal of groups to form double bond. So it's dealing with a double bond or reverse break the double bond or reversing the case of double bonds. Examples are decarboxylases and synthesases. So here the fumarase in the fumarate example, in the fumarate sample, so there is a double bond there, so it's breaking that double bond into single bond. So you have a hydroxyl group being attached here with the help of water, and other side H also. So there are two OH and H being added and the double bond is broken up with the help of lysases. Then isomerases, it's catalyze the intermolecular rearrangement. So like in epismerases, mutases. So in this case, phosphoglycerate mutase. So 3-phosphoglycerate is changing into 2-phosphoglycerate with the help of phosphoglycerate mutase. So you have your phosphate group to be attached here to the oxygen group, but it's then going to exchange to the second carbon onto the oxygen one and attached to there. So this is 3-phosphoglycerate, then 2-phosphoglycerate. It's changing the isomerization. Then comes the ligases, which catalyze the reactions in which there is a carbon-carbon bonds, carbon-sulfur bond, carbon-oxygen or carbon-nitrogen bond is made or broken. So either they are made or broken. So in this case, what we are showing is there's a DNA strand with the hydroxyl group and one DNA strand with the phosphate group. And with the help of DNA ligase, you have your DNA strand on both sides, and you have at the end your attached part of this. Your phosphate group is being attached very nicely. So your two DNA strands are attached with the help of your DNA ligase group. Further, in most cases, in the nomenclature, so we add the enzyme with the thing called AZ then. Uh, so most common name for hydrolyze is derived from the substrate, that is uh, urea. For in urea case, we just remove A, and instead of A, we replace it with the A's. Then we call it urease, the enzyme that is the common name for hydrolyze in the urea as a substrate. And in the case of lactose, we remove O's from it, and then we add A's at the end. So it is a lactase enzyme. So other enzymes, they also have the same name, but we add what their action they are doing, what the reaction they are catalyzing. So if they are lactate dehydrogenase, if they are like removing the hydrogen group from lactate group, then they are known as lactate dehydrogenase. If they are decarboxylase, so they are removing the carboxylic group from the pyruvate, then they are known as pyruvate decarboxylase. So some names are historical, they have no direct relationship with the substrate or reaction type. So they are just named like as it is, such as catalase, pepsin, chymotrypsin, trypsin. So these names are given to them. So just like that. So we have discussed about nomenclature, uh, various type of your enzymes, various types. So now comes a little bit about the activation energy, how it is working out. So here you can see K equivalent. This is a equilibrium constant, which has been produced by the reaction of there is A with the numerical substrate as A here and above, then B, then small b and above. So what you do is you write the product first, then with raised to power of its uh, small b, and then as a reactant, then raised to power of small a. So an enzyme speeds up a reaction by lowering the activation energy and changing the reaction pathway. So this provides a lower energy route for conversion of substrate to product. And every chemical reaction is characterized by equilibrium constant, that is KEQ, which is a reflection of difference in the energy between reactants and the product. So it's just telling us the how much there's a difference in energy between your reactants and products. Further, here you can see in A reaction, like in the normal without enzyme, this is an uncatalyzed reaction. So in which reactant A and B are joined and progress to the reaction. And this is the activation energy is required EA. Then they are making the product C plus D. And then in the energy reaction, what you're seeing is reactant with the enzyme part. It's decreasing the activation energy. It's decreasing it in a huge amount. So it doesn't have to go so much of energy required. So with the help of enzyme, it just needs a little bit of energy. And then easily your progressive reaction will be done and a product will be produced. So uncatalyzed reaction has large activation energy, we can see at the left, whereas the catalyzed reaction activation energy has been lowered significantly and increasing the rate of reaction. So that's it. What is the difference between the reactants and products? So effects of substrate concentration on enzyme catalyzed reaction. So there is a uncatalyzed reaction and enzyme catalyzed reaction. So uncatalyzed reaction, you can see that rate of reaction is almost proportional to the substrate concentration. So both are proportional to each other. So somebody's message. So both are proportional to each other. Whereas in the enzyme catalyzed reaction, there is a first is increasing rate by rate, then suddenly there is a bell-shaped curve, then they become constant afterwards. So that's what happened in the case of enzyme case where you have a Vmax and you have catalyzed reaction in that way. So you can see the rate of uncatalyzed reaction increases as the substrate concentration is increasing. So as the substrate is increasing, concentration is increasing, your rate of fraction is also increasing. So that's uncatalyzed reaction. But in the enzyme case, it happens in the two stages. The first stage is the formation of the enzyme substrate complex. First, your enzyme and your substrate, they are formed. Enzyme and substrate are formed, and then this is followed by the slow conversion to product. Then they are followed by the slow conversion to product. Rate is limited, uh, and enzyme availability. It depends upon the enzyme availability of that. And then enzyme substrate complex. So this is how the complex is shown to you. So first is enzyme substrate. In the first step, you have enzyme substrate complex is being made. Then it is a transition state in the step 2, where it is in the transition state is of enzyme. Then in the enzyme product complex in the step 3, and then step 4 is enzyme and product are made. So the first step involves ES, then ES transition state and enzyme product complex. So part of the enzyme that is combining the substrate is the active site, and active site characteristics includes pockets or cleft in the surface of the enzyme. Shape of active site is complementary to the shape of the substrate, and the enzyme attracts and holds the enzyme using the weak non-covalent interactions, and conformation of the active site determines the specificity of the enzyme. So these are all depending upon the enzyme substrate complex details, and this is um, yeah, just confirming the whatever we have studied so far. So lock and key enzyme model. So this is shown to you now. So this is enzyme and this is substrate and it is being attached to it. But in this case, there is one disadvantage, that is this is assumed to be a lock and key substrate model, but enzyme and substrates are made to fit exactly. But this model fails to take into account the protein conformational. There is some conformational changes are happening in the protein, which accommodates the substrate complex, and this was not told in this model. Then comes the induced fit enzyme model. So in this model, induced fit enzyme model, enzyme action assumes that enzyme active site is more flexible pocket. So enzyme feels that the active site is more flexible pocket whose conformation changes accommodate the substrate molecule. So it changes conformational positions, its structure. So earlier, if it is like this, like that, but it changes its shape according to its substrate. Whatever the substrate wants to get added in, it changes according to its shape itself. Then your enzyme substrate complex are attached to it. So transition state and product formation. So so how does this enzyme promote faster chemical reaction? So how your enzymes are promoting a faster chemical reaction? So this is as the substrate interacts with the enzyme, its shapes changes, and this is new shape is less energetically stable. So as the substrate is interacting with the enzyme, its shape changes. So with the less energetic stable, and transition state has features of both substrate and product. So transition state that is there, it has both the function of substrate and product. Yeah, it's on the both side and falls apart to yield product, and it's just destroy itself to become product, and which dissociate from the enzyme, and then the enzyme is dissociate afterwards from the its. So like E and P. So this is one example showing you of the same possible type of transition state changes. That is, you can see that there's a glucose molecule, it's a bit small, just go through that, and there's a fructose molecule. And then it is a sucrose molecule is made, water is removed. So they are both joined together. So how was this possible in this case? So this was possible because of the transition state. So you have your enzyme plus substrate, and this enzyme transition state ES complex was made, and then they were joined together. So both have OH group, and they joined together within the enzyme and giving rise to enzyme plus product at the end. So that's how your enzyme is giving you sucrose at the end. So enzyme might bring two reactions into close proximity and maintain the proper orientation. So this is how they maintain the orientation also, and also the shape of conformational changes are done in the enzyme, and hence you have your uh, product that you were looking for. Okay, what are the other possible types? It could also modify the pH of the microenvironment, donating or by accepting the H plus ion. So it could take also H plus ions or donate it that to the environment. So it could change the pH of our sample test also. So there is also something thing called cofactors and coenzyme. Here in this figure we can see that there is an apoenzyme and there is a substrate came to it, but as there is no cofactor, it's not activated. So the cofactors join in as a cuprous as a copper plus plus. So functional enzymes will be active binding site, and then you have a reaction occurring. So no enzyme substrate complex, no reaction in the first case, but in the second case, after adding the copper, you have an enzyme substrate complex and giving you to the reaction that you were looking for. So active enzymes are holoenzymes, polypeptide portion of enzymes, apoenzymes, whereas the non-protein prosthetic group are the cofactors. So cofactors are the bound to the enzyme for its maintain the correct configuration of the active site. So they are as metal lines, organic compounds, organometallic compounds. So all these could be attached to it as a cofactor. And this is another example of coenzyme is required by some enzymes. So they are the organic molecules bound to the enzyme by weak interactions and hydrogen bonds. Most coenzymes carry electrons or small groups, and many have modified vitamins in their structure. So here you can see is an enzyme with coenzyme positioned to react with two substrate. So this is your enzyme is, and here is your coenzyme, and to it there is substrate is attached to it. Then there is substrate 1, and to the substrate 1 there is a functional group is attached to it. So there are two substrate, one coenzyme, this is your enzyme, and functional group is attached to your substrate 1. So what will happen? Coenzyme picks up the functional group from substrate 1 and transfer to the substrate 2. So it took it from the first coenzyme and attached to the substrate 2. And the products are released with the product 2 having the functional group of your substrate 1, and substrate 1 is without the functional group. So that's how your coenzyme are also required, quite essential to carry the electron or small groups from one thing to another. So these are the various examples of various soluble vitamins and their coenzymes and their specific functions. So in vitamins there is a thiamine. The coenzyme is thiamine pyrophosphate, and the function is decarboxylation reactions. And vitamin is riboflavin, and the coenzyme is flavin mononucleotide and carrier of H atoms. Further, niacin has nicotiniamide like NAD plus, and pyridoxine as a B6 vitamin has pyridoxal phosphate. Cyanocobalamin, that is B12, has deoxydienosyl cobalamin. Folic has tetrahydrofolic acids, and whereas a pantothenic acid has a coenzyme A, biotin has biositine, ascorbic acid is it's still not known what is the name, but it's still they have coenzyme also, which is doing the hydrolyzation of the proline, lysine, and collagens. Our pug. So let me just make this sound a bit away. So, students, let's continue. Allosteric enzyme in metabolism. So, the third reaction of glycolysis, placing a second phosphate on the fructose 6 phosphate. So, here's in the fructose 6 phosphate, you have a phosphate group attached to it, and you have ATP to it. And then, with the help of the enzyme phosphofructokinase, what does it doing? It's attaching a one more phosphate group. It's making fructose 1,6 biphosphate like this. So that is the function of allosteric enzyme. So ATP here is acting as a negative effector and AMP is a positive effector. of the enzyme phosphofructokinase. So ATP here is a negative. If I'm adding AMP, that will be a positive effector to it. So it doesn't let the reaction go further. So how this feedback inhibition works? So allosteric enzymes are the basis of feedback inhibition actually. So with the feedback inhibition, a product late in a series of enzyme catalyzed reaction act as a service of as an inhibitor for previous allosteric enzymes early in the series. So in this case, so if it is E1 is our enzyme, it's producing a product B, then with the help of enzyme E2, then C, and further on like it go to F product. So F in this example could serve to inhibit the activity of enzyme E1. So this F product will be inhibiting the activity of enzyme E1, and product F act as a negative allosteric effector on one of the early enzymes in the pathway. So this is how your reaction is working. So product F act as a negative allosteric factor on the early enzyme in the pathway to the E1. So that's how the feedback inhibition is working in this case. So now what are proenzymes? Proenzyme are the one which are in the inactive form, and it is converted to active form by either proteolysis, that is hydrolysis of the enzyme, or when needed at the end of the or when needed at the active site in the cell. So pepsinogen is synthesized and transported to the stomach where it is converted to the pepsin. So these are the various enzymes of our digestive tract written here. That is proallistase, which is giving activator trypsin, trypsinogen, which activator is trypsin and enzyme is trypsin, chymotrypsinogen, pepsinogen, pro-carboxylic peptidase, where activator is trypsin and carboxylic peptidase A. So these are the various examples of proenzymes of the digestive tract. Further, to see the protein modification, in protein modification, chemical group is covalently added to or removed from the protein. Covalent modifications either activates or turn off the enzymes. The most common form of protein modification is addition or removal of the phosphate group, and this group is located at the R group with free OH group, that is serine, threonine, and tyrosine. So that was about how there could be different modification of proteins we are talking about. Now comes the part about to talk about inhibition of enzyme activity. So enzyme could be inhibited. Chemicals can bind to enzyme, eliminate or drastically reduce catalytic activity. Classify enzyme inhibitors on the basis of reversibility and competition are. So irreversible inhibitors bind tightly to the enzyme and thereby prevent formation of the ES complex. So reversible competitive inhibitors often structurally resemble the substrate and bind at the normal active site. And this reversible non-competitive inhibitors usually bind at some place other than the active site, and binding is weak and thus inhibition is reversible. So further, irreversible inhibitors could bind very tightly to the enzymes. So binding of these enzymes to one of the R group of amino acids in the activity site. So this binding may block the active site so that enzyme substrate complex cannot form. So further, irreversible inhibitors includes arsenic, snake venom, nerve gas. So these are the various example of your irreversible inhibitors. So there are also reversible competitive inhibitors. So they could reverse, and competitive enzyme inhibitors are also called as structural analogs. So they are the molecules that resemble the structure and charge distribution of natural substance of the enzyme, and also the charge distribution of the natural substance for an enzyme. So resemblance permits the inhibitor to occupy the enzyme active site. So once inhibitor is at the active site, once it is attached to the active site, no reaction can occur and enzyme activity is inhibited in that case. So inhibition is competitive because of the inhibitor and substrate compete for the binding to the active site. So degree of inhibition depend upon the relative concentration of enzyme and inhibitor. After reversible competitive inhibitor, there is non-competitive and reversible inhibitors. That is, they are the bind to the R group of amino acids or the metal ion cofactor. Binding is weak and activity is restored when inhibitor dissociate from the enzyme inhibitor complex. So these inhibitors do not bind to the active site, do modify the shape of the active site once bound elsewhere in the structure. So proteolytic enzymes cleave the peptide bond in proteins. So these enzymes break the peptide bonds that maintain the primary protein structure. These enzymes specifically bind on the hydrophobic pocket, a cluster of hydrophobic amino acids brought together with 3D folding of protein chain, and chymotrypsin cleaves the peptide bond at the carboxylic end. So here you can see end of methionine, tyrosine, tryptophan, and phenylalanine. So these proteolytic enzymes, they are cleaving the peptide bond. So here in this case, alanine and phenylalanine and glycine, they are attached together. So they are breaking the peptide bondage like this here. So what are the applications of these enzymes in medicines? So they are the diagnostic enzyme levels that altered with the disease, such as in a heart attack cases, lactate dehydrogenase, creatine phosphate, serum glutamate, oxalate acetate transminase, SGOT. So they are the we check. So their enzyme activity is changed in the heart attack cases. So whenever you go to the in a group also in a heart attack also, so you can check all these parameters to that. So pancreatitis, amylase and lipase are being used also. Analytical reagents, so enzyme used to measure another substance such as urea converted to ammonia via urease or blood urea nitrogen by measures. Replacement therapies are done, genetically engineered beta glucose cerebrosidase for Gaucher disease are being used. So I think this is not for you. This is the references. So you can check these books that we have made for these lectures from. Thank you very much. You can contact me on this email ID if you have any things to inquire about. I will be happy to do so. So these are the end of your slides. Let's continue the next part now. So we had an intensive talk about DNA yesterday with the various videos, but I want to take you to the subjective part of it because it's good to watch many videos and to understand uh, various parameters, how DNA is working, how it is double helix, how adenosine is attached to tyrosine, how glycine is attached to cytosine, and making a double helix structure with a major groove and minor groove, and how it is different from the RNA with the deoxyribonucleic acid and on ribonucleic acid. So we all know about DNA and RNA, what are the differences, but what are the various types of DNA, how the repair system works, how it's dealing uh, with the things of yeah uh, other concept of it. So I will be dealing in this case with that part now in the next lecture. So uh, any questions so far? But the record sessions will be not good because it is going via Google Meet and YouTube is going on together. So I don't have that big bandwidth internet at house so that I could run two live stream also and Google Meet also. So let's see if I can send you the link here. So later you can copy it also in the group. Yeah, it's being copied. Just check it yourself, or just I send it there only. But this will be not good enough recorded for you. Just keep it with yourself. Don't share with anyone. Just this is just for you for your workshop. After watching it, keep it within you. Good, not so. Biochemistry, DNA and its structure, functions, and types, mode of replications, and pair. So I will skip most of the initial part because we have discussed in the video. So just go through them by them myself for you. It's a quite subjective. So I hope you can clear that on your own. But still, wherever things have to be told to you, I will speak about it. So So this is some history about DNA, how it was being found, how there were a lot of research was going on. So all about various bizarre things were happening at that time, where the 3D dimensional structures were finding out, genes were being finding out, nucleotides were finding out, how they are complementary to each other, how DNA is complicated to that with other things. So that was being done at that time. Then comes the Watson, Crick, and Wilkins. These were the young Swiss physicians. Also, Friedrich Miescher isolated something no one had ever discovered before from the nuclei of the cells. They called it at that time as nuclein, and this is today called as nucleic acid or Na. That is in DNA and RNA. It is present. Then Gregor Mendel did a lot of observation in the pea plant to check how these nuclein are connected to his experiments. Further, for a long time, in the connection between nucleic acid and gene was not known, but in 1944, the American scientist Oswald Avery managed to transfer the ability to cause disease from one strain to another. So the Avery experiments, they're quite famous one. So they did it to check that the reason was for the generation of disease was the nucleic acids. So in the 40s, the puzzle was solved, and it was so many skepticals were there about it. But DNA, including four bases: adenine, thymine, guanine, and cytosine. So these four A to ATGC, but nobody had the slightest idea of what the molecule might look like. So in order to solve the elusive structure of DNA, a couple of distinct pieces of information needed to be put together. One was that the phosphate backbone was on the outside with the bases on the inside. Another was the molecule was a double helix. So these were the initial research was happening. Then the James Watson came from United States with the Francis to work with Francis and Crick, and they work in the University of Cambridge. So Crick was already using the process of X-ray crystallography to study the structure of protein molecules. So together, Watson and Crick use X-ray crystallography to produce Rosenfeld, Franklin, and Morris Wilkins at the King's College London to decipher DNA structure. So out of all this university, for myself, I have worked at the King's College London for a couple of, not worked, but I had a collaboration with them, with the big scientist, where my work was being done at the other lab also. So I had a little bit chance of meeting the great scientist during my PhD time. So this is what they already know, that the Watson Crick, the scientists of DNA molecule, that DNA is made up of subunits, which scientists call as nucleotides, and each nucleotide is made up of a sugar, phosphate, and base, and they are as known as adenine, cytosine, guanine, and thymine. Further, so purines and pyrimidines were equal in amount. Adenine and thymine are equal in amount. Guanine and cytosine are equal in amount. And a basic structure of DNA molecule is helical, with the bases being stacked upon top of each other. Just a second. Let me have a sip of water. So this we have done. I will not repeat all these things. So A is for G is for guanine, C for cytosine, thymine. There are purine bases, pyrimidine bases. This we all did. So this is the 3D structure of them actually, thymine and cytosine. Students, somebody's mic is unmuted. Just mute yourself, please. So then how your deoxyribose sugar is playing role, as a this is shown as a ball stick overview of that, and with the frame view also. Deoxyribose sugar has five carbons and three oxygen atoms, with the carbon atoms numbered as 1 2 3 4 5. So we all know about the structure of it. The nucleosides we know is just including your sugar group and nucleotide group. Sorry, your base pair. So they together make your nucleosides, and nucleotides including the phosphate groups attached to it. Then they are making together your nucleotides to it. So somebody please unmute yourself. I'm listening autos sounds. I don't know who is. Kosik Vijay, please. Kosik, could you please unmute yourself? Great, thank you so much. It happens sometimes. Uh, no worries. Um, then this is the backbone how your base pairs, pyrimidine, purines, they're being attached to the phosphate group with the glycosidic bonds and being arranged in this way. Phosphodiester links, sorry, between deoxyribose sugars. So alternating backbone. So on the other side, you have alternating base pairs to it and being attached like this 3D structure. So you have base, hydrogen bond in between, and a sugar phosphate backbone also. So that's how somebody wrote, I think. Okay, nobody. So I thought somebody asked me. So this is how your basic DNA works like. I will not go in detail of this. Please go through. So this is mainly text for you. I have created. It's done from the famous biology background books about basically from the genetics books. So just go through them, read about it, because we have extensive studies about them already yesterday, and we know that they're anti-parallel and how they are moving from 5 prime to 3 prime end. So I don't want to go in again repetition of that, but I kept these lectures for you only, for you students only, so that whenever you have a subjective exam, when it comes to write in a good amount, so you should have some material for you. So this is good for your examination point of view. Any examination comes related to your DNA point of view. That's why these lectures are so filled with the words here. So don't worry, some slides are like this. So these are the various features of double helix structure. So I could repeat some. Diameter of helix is 20 angstrom. Their helix can virtually be any length when fully stretched. 33.4 angstrom is the separate planes in which the adjacent base pairs are located. Double helix structure is a chemical subunit. It has two aspects, that is in which these chemical subunits are arranged to form long chain molecules. So you have phosphate bases. The same things are being repeated. So what are the functions now of your DNA? Mainly it's carry the genetic information of your cell and consists thousands of genes, and the flow of information is done from the genes determining the protein composition and thereby function of your cell. So DNA is situated in the nucleus, organized into chromosome, whereas each cell contain the genetic information, and DNA is therefore duplicated before the cell divides. So when proteins are needed, corresponding genes are transcribed into RNA, and then RNA is then first processed so that non-coding parts are removed. Is then transported to the nucleus, and outside the nucleus the protein are built upon the codo in the RNA, that is your translation. So this is how DNA is being used in the replication, transcription and translation, and transportation and processing of a RNA. So that's how the DNA has a main role in it, quite important in our functioning of our things. So DNA can be classified in various ways: number of base pairs per ton, calling pattern, location structure, nucleotide sequence, a number of strands. So number of base per turn is depending upon the nucleotide base per turn of the helix, tilt of the base pair, humidity of the sample, and the DNA can be observed in four different forms, namely A, B, and C and D. So calling pattern, whereas in the calling pattern, it could be of right-handed and left-handed type. Most of the DNA molecules are right-handed, that is calling of the helix in the right direction. and it is called positive coiling. Whereas the all forms of DNA, that is A, B, C, D, are right-handed. The Z DNA is the only one which is has a left-handed thing. So this is like right-handed coiling. So if you can make like this, right-handed, and if this will be A, B, C, D, where the Z will be on the left-handed. It's a left-handed double helix structure going in the anti-clockwise. And DNA is considered to be associated with the gene regulation. Based on location, DNA could be it's of three types, that is chromosomal DNA, cytoplasm DNA, and promiscuous DNA. The chromosomal DNA is found in chromosomes and are called as chromosomal DNA or nuclear DNA. Whereas the cytoplasmic DNA is found in the cytoplasm, especially in the mitochondria and chloroplast. Such DNA is important in the cytoplasmic inheritance and has a circular structure. Whereas the promiscuous DNA, as name suggests, is something with the found in also chloroplast, mitochondria, nucleus. It's present in everywhere. This suggests that some DNA sequence move from one organelle to the other. Such DNA is referred to as promiscuous DNA. Then comes the structure of RNA. So we have discussed about the function of DNA, various types of DNA, and how the function works according to the type of it. Now comes the structure of RNA. So mainly the difference of between DNA and RNA is a ribose sugar. It has complete not deoxy one, but complete ribose sugar, nitrogen bases, and phosphate groups. The nitrogen bases includes adenine, guanine, cytosine, and uracil. Sorry. In DNA, thymine is present in place of uracil, and deoxyribose sugar is found in place of ribose sugar. So in RNA, the pairing occurs between adenine and uracil, and it is usually single-stranded. However, some viruses could be double-stranded RNA. So within the DNA, so as we are discussing about other form, so this is something telling about you again, A, B, C, right-handed helix, and Z is the left-handed helix, and B is the major form that is found in the cells, whereas Z is found in under high salt conditions, but the cellular environment is actually so low environment. So several features have also found zDNA to be present in the low salt environment also. So that's the basic difference between DNA and RNA. That is, strands are usually two or rarely one. In RNA, it's mainly one. Sugars is deoxyribose sugar, that is ribose sugar. Your bases are like that, and pairing is ATGC, but here is AU. Allocation is present mostly in chromosome or in mitochondria and chloroplast, and that is present in the chromosomes and ribosomes. So further mode of replication, there are three possible modes of replication, that is dispersive, conservative, and semi-conservative. Uh, this part I will not touch because this will be more, we will be going hardcore of DNA then. So this I will skip because I have to teach you a lot of other things also. So just go through them. We all know that there is called yeah, this Okazaki's fragments, that's a different part of it. So that things just go through them with the unwinding of your DNA, then winding of DNA with a 5 prime to 3 prime end, then it's opening of it, then we having new complementary DNA being attached to it, then it's producing a new sequence from the 3 prime to 5 prime end and leading to your new DNA synthesis to the complementary DNA. So that's how DNA replication is done. Then DNA base pair, so damage could be done to DNA. DNA repair system is about to take care of DNA repair system. So various damages are grouped into following distortion. So single base pair changes means that some single base, example deamination is done of a 5-methyl cytosine result in thymine, or by covalent addition of small group to base which affects its pairing behavior. Structural distortion, these changes generally adversely affect the replication of or transcription of the affected DNA molecule. So they are presented by single strand neck or removal of base, covalent links, all are dealt in the structural distortions. Examples are pyrimidine dimers, addition of bulky adducts to a base which may distort the configuration of the double helix. So DNA repair system recognize a variety of changes in DNA to initiate actions. So these are as follows. That is direct repair system, excision repair system, mismatch repair system, tolerance repair system, and retrieval repair system. So a little bit about direct repair system. It's a reversible or simple removal of the damage to the DNA is known as a direct repair system. Example, removal of the covalent bonds between the 2 or 4 carbon atoms of the two thymine residues participating in the formation of thymine dimers. So they are making thymine dimers also. The thymine dimers are generally formed due to the UV radiations. So TT groups are being made, therefore interfere with the replication and transcription. So if your TT are made, though it's very difficult to make to do your transcription and also translation afterwards. So replication is not possible. All things will be not good and done. So enzyme can bind to thymine dimers in the dark, but requires energy from the blue light of removal of the covalent bonds. So that's process. It's known as also photoreactivation. So direct repair system is widespread in nature and especially important in the plants also. So excision repair system in the DNA repair system is this is done by the following steps. One is the recognition of the incision. First, it recognizes the where is the by endonuclease has been done. Then this enzyme can cut the effective strand on both of the enzyme of it. Then excision. After the incision, excision is done 5' to 3' with the exonuclease is digested away with mispaired section, generate a single strand region in the DNA double helix structure. Then the synthesis step is done later on. Then in the mismatch pair, when the single bases in DNA are mismatched, either due to the alteration in the existing bases or due to the early replication or the structure or distortion of double helix. And the last and the final one is the retrieval system. These systems known as the post-replication repair or a combination repair. So that's it. That was done from the DNA. Its various functions, types, and how your repair system is work. Uh, so So whatever that I have showed you is the advanced version of DNA, which you might be not learning anywhere else. So whatever studies that we are doing um for the good cause actually, so that you are aware that the and they are quite important actually. All these systems, which are not taught anywhere. So this is exclusively being taught you for the first time about various type of DNA structural and in the subjective manner of it. So so now something about RNA. We have discussed about DNA, but there are three types of RNA. That is your ribosomal RNA, uh, then messenger RNA, and then is a tRNA, transfer RNA. So we will discuss little bit about all of them, how they are important, how they are working out, and where and how they will be working for us. So this RNA molecule is different from RNA from DNA in having the ribose sugar. That is the most common nitrogenous basis of RNA. That is DNA, guanine, cytosine, and uracil. The pyrimidine uracil substitute the thymine of DNA in regions where purine pyrimidine pairing takes place. Adenine pairs with uracil, guanine with the cytosine. In addition to the four bases mentioned above, RNA also have unusual base pairs. In the unfolded region of the base pairs have no complements. Because of this, RNA does not have purine pyrimidine equality that is found in the RNA. So this is the chemical structure of RNA. So you have a phosphate group. These are the bases like guanine is being attached, and then you have a phosphate with the other with another sugar attached to it. So this is how RNA structure mostly looks like. Uh, Chemical structure of RNA. It's a secondary structure that is a stem loop and hairpin. So there's a band between bond being between here in the stem style here, and in hairpin there's a bonds happening here. So they are usually bases in they are unusual bases in RNA than the DNA. And all normal RNA chains either will start with adenine or guanine, and three types of cellular RNA have been distinguished. So they are messenger RNA, ribosomal RNA, and soluble or transfer RNA. So there are three types of RNA are there. Uh, so ribosomal and transfer RNA comprise about 98% of all RNA, and all three forms of RNA are made on DNA template. And transfer RNA and messenger RNA are synthesized on DNA templates of the chromosomes, while ribosomal RNA is derived from the nucleolar DNA. And okay, most of the RNA synthesized during the cleavage is mRNA, and synthesis of transfer RNA occurs at the end of cleavage, or ribosomal RNA synthesis during the gastrulation. So that's the major difference between your DNA and RNA looks like. DNA is usually genetic material, where RNA is usually your uh, genetic material of some viruses. So pentose DNA is a pentose sugar deoxyribose, whereas RNA is a pentose sugar. So these are the various differences. Just go through them. There are like 14 differences written there, which is quite important for you to remember from the examination point of view. So just keep in mind these things. Then ribosomal RNA, is the name suggests, it is found in the ribosomes. So it comprises about 80% of the total RNA of your ribosomal of your total RNA. And the base sequence of ribosomal RNA is complementary to that of region of DNA where it is synthesized. In eukaryotic, ribosomes are formed on the nucleolus, and ribosomal RNA is formed from only from the small section of DNA molecules. Hence, there is no definite base relationship between ribosomal RNA and DNA as a whole. So there is not so much difference between these two. Further, ribosomal RNA consists of a single stranded twisted upon itself in some regions, and also helical, connected by the intervening single stranded regions. In the helical region, most of the base pairs are complementary and are joined by the hydrogen bonds. In the unfolded single stranded regions, the bases have no complements. Ribosomal RNA contains the four major RNA bases with a slight degree of methylation, and shows the differences in the relative proportion of the bases between the species. So this is how it's looked like. Your ribosomal RNA, your structure is, so it is sometimes unopened, like there's not they are not binded. Then they are started to have a binding of it. Then they make helical structure a little bit. Then they still again open. So you have little bit of base pairing AUGC, CGUAAUGC. So we have a little bit of it, but somehow it's open. So ribosomal RNA does not show purine pyrimidine inequality. The ribosomal RNA unfolds upon heating and refolding protein cooling, and it is stable for at least two generations. And ribosomal RNA consists of proteins and RNA. So 70S uh, ribosomal RNA prokaryote consists of 30S subunits and 50S subunits. Yeah, and whereas the 30S subunits contain 60. These are Sydbuck units, Sydbuck coefficient. These are S Sydbuck coefficient. So 16S S ribosomal RNA, while 50S has 23 and 5S ribosomal RNA. So 80S eukaryotic ribosome consists of 40S and 60S subunits. In vertebrates, the 40S subunit contains the 18S ribosomal RNA, while 60S contains the 28 and 29. So this is goes like this. So there are three types of ribosomal RNA based on the sedimentation we have discussed. Molecular weight. Two of these classes are high molecular weight RNA, while the third is a low molecular weight RNA. So the high molecular weight RNA with over a million, 21S and 29S RNA, and high molecular weight with RNA with a molecular weight with below 1 million, they are around 12A to 188 low molecular RNA. So So ribosomal RNA, it's looks like this. You can see very nicely uh, that there is a 40s unit up and 60s unit below, right? And this is the small subunit, and this is the larger subunit. Uh, and then there is a groove inside this, and here your tRNA is being attached to it. And there's a tunnel with the polypeptides being produced with the various amino acids, and it is attached to the endoplasmic reticulum. And this tRNA is attached to the mRNA here, and this tRNA is attached to the anticodon and giving rise to your mRNA, your amino acids polypeptide chain coming out of it. That's we have saw the video yesterday. So whatever we are studying here is the one from the video. So go through those videos also once more time to take care of your home message from these slides also, so that you can combine them together also later. So the reason that I showed you all the videos first to clear your concepts, then bringing the subjective knowledge, because that's what happened. We understand the concept. Everything in the books is written in a very, it's in 2D form. But once you see the things happening in with your naked eyes, that okay, so ribosomal RNA is making the proteins with the help of tRNA and mRNA in this way while standing on the endoplasmic reticulum, and things are going in that way to produce your translation later on. So this is how your translation is being done. So that's the main purpose of all this. So then we are done with the ribosomal RNA now. Now comes the messenger RNA. So Jacob and Monod, they proposed the name messenger RNA for the RNA carrying information for protein synthesis from the DNA to the sites of protein information. So it consists of 3 to 5% of total cellular RNA. Size of messenger RNA is around 500,000 is the average size, and sediment coefficient is 8s. And stability, in the term of stability, the cell does not contain large quantities of mRNA, and this is because mRNA, unlike other RNA, is constantly undergoing breakdown. Further, under your mRNA, it has following structural features. It has a cap, it has non-coding regions, it has initiation code and coding regions. So these are the various features of it, of messenger RNA. Then comes your transfer RNA, which is around 10 to 20% of your total RNA, having a molecular weight of around 25,000 to 300,000 of your transfer RNA of it. And structure of transfer RNA of this structure was first worked out by Holy et al. For yeast alanine transfer RNA, which has 75 different transfer RNA work for them. So he called that model at the first time that he found as clover leaf model. It looks like a leaf model for Holy, which was widely accepted at that time. So this helps in the protein synthesis reaction, and there are around 20 different kinds of tRNA which serve as a carrier of specific amino acids to the translation. They are L-shaped. The amino acid is attached to one end of other and consists of three anticodon nucleotides. The anticodon pairs with the codon in the messenger RNA, ensuring the correct amino acid is attached to it. The L-shaped tRNA is formed from the single-stranded RNA that folds into proper conformation, giving rise to four different regions of double-stranded RNA and formed during the folding process. So this is how your transfer RNA looks like, the main structure. You have T size C arm here, acceptor stem here, D arm here, and variable arm on this side. And there is an anticodon arm here, which is attached to the RNA, mRNA side. So the two ends of the molecules are acceptor stem regions where the amino acid is attached. So acceptor stem region, where this is where your 5' to 3' end is written. Your amino acids are attached to it. This is called your anticodon. And another side is the anticodon is exposed to the end of the anticodon arm. So this is anticodon arm where your anticodons are attached to it. Then the two other stem loop structures are named after modified nucleotides that are found in these parts of molecule. That is D-arm contains dihydrodeulate residues, that is TYC-arm, which is, and also it has ribothiamidylate residues, and so pseudo urodylate residues and citylate residues in that order. So that is how T pseudo and citylate names came actually. So then there is a variable arm also, which is variable as per the expectations. So they are barely noticeable, while in others it is largest arm. So tRNA are usually drawn in the cloverleaf form, emphasize the base pairs into the secondary structure. So this is the transfer RNA, tRNA or soluble RNA shown to you. The classification of tRNA study, that is different tRNA show the structure of the receptor stem and anticodon arm, and T signs are constant. So according to the three classes of tRNA has been recognized according to the variable region. So it's a class 1 where you have D4, V4, 5 with the 4 base pairs in the D stem and 4 to 5 bases in the variable loop. And then there is a D3, V4, 5 which has 3 base pairs with D stem and 4 to 5 base pairs in the variable loop. And the D3 Vn is with have the base pairs of the D stem and the large variable uh, arm in that. So this is how your transfer RNA is being separated according to its model. So this is by that scientist, cloverleaf model is being recognized at the bottom. You can see it. That is, you have a carrier end, you have enzyme site, ribosome sites, and you have codon site here. So this is all they are being attached and nicely done to your for your experiments, for your further translation actually. Then in the tRNA, there is an initiator transfer RNA that is known as tRNA, and its name is methionine, which like in to initiate experiments. So it is methionine in the case of eukaryotes, and in the case of prokaryotes, it's N-formyl methionine, respectively. So they are quite the specificity, they are quite specified transfer RNA. So two important steps in the translation during the protein synthesis are the activation of amino acids and transfer of amino acids to tRNA. Each amino acid has specific activating enzyme, that is tRNA amino acyl synthetase. Remember this come quite number of time in exams. And there are 20 different tRNA amino syntheses are there for 20 amino acids in the proteins. So thank you very much on this. So that was quite complex, I have to admit. For you might be, for some might be easy for someone, because this is the basic, this is the most simplified version of the slides that I could make for you to understand you between the ribosomal RNA, messenger RNA, and tRNA, and how their various functions are, how their various types are, and how their applications looks like. So this was my initiative to touch you the biochemistry part of, little bit biochemistry part of DNA and RNA. So how they are being done from the chemical perspective, how their structure is working like that, why they are because in the videos we just saw that how easily everything is done, but when we come at the molecular level, when we see at the biochemistry point of view, when the reactions are happening, so this is how they get complicated. So that's it for this perspective. uh, so any questions so far? So I hope you are able to understand these things, because if you're not able to understand with the basic DNA and RNA structures and how their various types are working with the various forms of DNA and RNA, it will be uh, yeah, not so good factor afterwards. So I hope you are able to get in what being taught here. It's a advanced biochemistry, I could say, but this is part of your course in this case. So any questions? Okay, so uh, I I need a little bit water now. I take 5 minutes break. Then we initiate the next part of it with the various disease part also. And what would you suggest, students, that I have this part also of transcription, translation, and genetic code of protein synthesis. So this is the most easy version. So there's not much to told you about. This is uh, so this is all self-explanatory. So this we have done with the genetic codes of UCAG of our to make your table of UCAG, that you make UCAG on the horizontal line, and on the row you also make vertical and UCAG, and on the right side you make UCAG, and then this is how you make genetic codes for your translation steps. So UUC, UUC, then UUA, like that. So, and that's how you can create many genetic code for your reactions. So I hope you are able to understand, or just tell me that these, shall I teach you transcription and translation also with the figures point of view, or you can go through them on your own? Just I need your yes or no. So I will continue according to that, because this is for you. All these things are being done for you. Okay. So if so, just give me 5 minutes. I get back to you. Then we start with the next lecture uh, with the jaundice. Okay, I'm coming in 5 minutes now. Just give me a second. So, students, I'm back. So, now we will come to the medicinal part of our workshop, medicinal biochemistry. Nitish Kumar. Bache, the topic is just it's telling us about transcription and translation, how it is done with the help of the videos and how from DNA to mRNA and from mRNA to uh, your protein is being made with the help of your tRNA, ribosomal RNA, messenger RNA, how they are all working. Maybe I can show you some video, some images to explain it, but I will not go in detail with that because that will be too much for you all to absorb also. So, so this is at the first Mendel identify that DNA sequence, fluorochloro flower color gene having different colors being was because of the DNA sequence. Every flower having a different colors because of some gene was the reason behind it. So that's why so there's a central dogma to the life which says that DNA is making your mRNA, and from mRNA, your three codes, like this is your anticodons, they're making your polypeptide. So methionine, the initiator code, is producing AUG, then isoleucine, AUC, then serine, UCG. So like this, and then the stop codon, UAAA. Then your translation is stopped. So basically, your all these chapters are telling you about this. So that's how the gene expression works. So this is your DNA and how in the case of transcription is working. You have your DNA. DNA is a coding strand is there. Then it's unwind the DNA. Then from there your mRNA is produced. Instead of A, it's being attached to the by U in this case. So no T will be there. and you will have a separate template of RNA that will be your primary transcript here in this case. So in humans and in bacteria is different somehow on the basis of that it is happening in the nucleus everything, but here in bacteria because there is no nucleus, it's their DNA is uncoiled and all open, so it's happening in there in that case. So here your mRNA is produced from the nucleus, transcription is done, mRNA is done, then it's came out of the nucleus, then the translation is happening with the help of ribosome, leading to the production of polypeptides chain of your proteins, and this is known as your translation. Whereas in the bacterium, you have your polypeptide transcription is done. From there, the mRNA is produced, and it just goes to the ribosome to produce your polypeptide chain of proteins. Then the translation is happening. So that was it's all about. So this is your genetic code is working out. AUG, AUC, UCG, they are having different codes for polypeptide chains. It's written as it there: methionine, isoleucine, and serine, and then stop codon afterwards. Then comes your steps of translation. So this is how translation is working out. This is your ribosome, and this is your messenger RNA with the codons, and the anticodon tRNA UAG has been attached to it with respect to then isoleucine is being there. Then the next part, the serine is coming being attached to it, AGC to UCG. Then they are being attached to another polypeptide chain, and this is how your mainly your translation is working in the protein case. Then comes the steps of translations. So there are many transfer produced around in a cell, but only a transfer RNA that matches the codon currently being read can bind and deliver its amino acids. So once a tRNA is snugly around its matching DNA in ribosome, its amino acids will add it into the end of polypeptide chain. So here is your matching of your DNA is going on. So you can see UCG being attached to AGC, and isoleucine is attached to serine, and then it's removing the further. So UAG is removed, AGC there. So next codon is coming here to read it, and this is how your steps of translation is working out. So there's a start codon and stop codon. Now we already know AUG and UA. So these are various codons that they're working start and stop. And this is the genetic code table that I've already explained you, that how it is done with UUU and UCU like this. And these are all together. If you make them together, so these are all coding for serine. These are all coding for proline. These are coding for threonine, alanine, valine. AUG is for methionine, that is initiator code, isoleucine, leucine, histidine, glycine, aspartic acid, glycine, arginine, glutamic acid, glycine, arginine, serine, uh, the both are for arginine also. So there are more of that. So you can create these like C, G, and A, like from three letters you can create. This is how all they are made. So you have to also, it is good if you could remember. It's good for your own studies and good for your things also. uh, Further, you can see there is a frame 1, frame 2, and frame 3. So frame 1 includes your mRNA. So this is the same way like we discussed. The reading frame it is working on. So it's keep on going like that. So there's nothing to discuss here. This is a diagram of your nucleotides, a very nice colored picture. If you want to understand more about DNA and RNA, go for this diagram. So it has separated this diagram giving you all the summary of that we have discussed so far. It is telling us the various pictures, various structures of pyrimidines, purines, and how they are attached to the ribose sugar, pyrimidines and pyrimidines, and with the phosphate group. And this sugar could be of two types, deoxy and ribose. and it's telling us also about which prime end which is being attached. So one prime end is your pyrimidines and purines are being attached, the base pairs. This is 2 prime carbon end, 3 prime carbon end, 4 prime carbon end, 5 prime carbon end. Your phosphate groups are being attached. So this is very nice summary telling you about the whole structure of it. Sugars, phosphate, properties of DNA. So 5 prime, this is all replication. So we also done with how messenger RNA we done that. It's working in such a nice way. tRNA we have discussed in details. So this is all very nice lectures. Just so for transcription, in the stages of transcription, you have a RNA polymerase actually that is helping you to produce mRNA from your DNA by uncalling it. So that's how it is important in this case. So there is initiation factor is there. In initiation factor, you have your RNA polymerase, and RNA polymerase is being attached, leading to the unwinding of your DNA, making a single stranded template. Then from the single stranded template, your mRNA is being produced. So first is the initiation, then is the elongation, then comes the termination. Then this you can see a hairpin shaped RNA is being produced after the termination of your transcription reaction. So there are eukaryotic RNA modifications being shown you. So there are exons and introns. Exons are one which are giving the reading for your things. So they are removed, so they are spliced, and you have mature mRNAs left afterwards. So translation, then comes the translation. So translations, as we already discussed, how it is done with the there is an initiation chapter is there. So this diagram is in more details from the biochemistry perspective. So first is your tRNA initiator. tRNA is being attached to the mRNA with the methionine. Then comes the next part of it, that is elongation. So in elongation, next part come, they join it, and then they make the next part. So this way, then the termination is happening after the elongation. Then that's it. So that was just a quite brief summary about transcription and translation for you, so that you could understand that. So I hope things were quite easy, was not that difficult. Great. So I was at, oh, maybe I should teach you first hypothyroidism. Anyhow, we start with this. So basically, jaundice is happening because of the bilirubin production in the liver from by the liver. When it is in low in production, our cell pigmentation heme is producing yellow color, and the skin is all yellow. So the kid is born within 24 hours. In the kids, it's happening. They are produced with a defect in the production of bilirubins. So their all bodies in yellow in color, and we see these defects in them then. So we will understand about introduction to jaundice in this lecture, and we understand the classification and evolution of the jaundice, and to study various approaches for the treatment to it. So introduction is jaundice. That is, it's a French word, jaune, yellow. Jaune means yellow. So J in French, it's become ya actually. So that's why we become jaune, yellow. So So it's a yellowish discoloration of tissue resulting from the deposition of bilirubin. Bilirubin is being deposited a lot, and that's why we have this jaundice. And tissue deposition of bilirubin occurs only in the presence of serum. Hyperbilirubemia is a sign of either liver disease or hemolytic disease. So in the introduction, keratinema and use of drug quinacarine and a few others causes of yellow coloration of tissue, and keratinemia can be distinguished from jaundice by a sparing of celeria. So in 4 to 37% patients treated with the quinacarine, yellowish discoloration of skin is seen. Unlike keratinemia, quinine can cause discoloration of scleria. So these are the various things could happen. So under steady condition and normal condition, the metabolic conditions, so bilirubin concentration is about 3.9 milligram per kilogram, or 250 to 350 milligram. It is produced by the breakdown of heme from RBC, spleen and liver. And 1 gram of hemoglobin yields 36.2 milligram of bilirubin. So this is a table figure showing us how bilirubin is playing role in the production of our human body functioning, production of heme cells also, how they are playing a role. So this is your macrophage here on the left side, in spleen, liver, or red bone marrow. So this is present in your macrophage. So they are red blood cells, death, and phagocytosis. So your red blood cells are dead, and what is happening is heme is produced, and then biliverdin is produced. Then further, uh, and also globins are produced. Globins are producing amino acids, which could be reused for the protein synthesis. On the other hand, from the heme, they are producing ferrocyanins, that is transferrin, and this one will do go to the liver and lead to the production of ferritin, and this ferritin, from ferritin with the help of enzyme transferrin and iron, it's producing Fe3 plus transferrin, and this Fe3 plus transferrin will go into the erythropoiesis in the red bone marrow and lead to the Fe3 plus globulin plus vitamin B12 and erythropoietin. All these come together and they do the production of RBCs. And the circulation of RBCs death and coming back, and it goes like for 120 days. It keeps going like for 120 days, 7, and this one. Then next part is uh, and the bilirubin is also produced by the biliverdin, and bilirubin going into the liver with the bilirubin is producing in the small intestine. Then from there bilirubin is producing urobilinogen, and urobilinogens are producing feces, and further they are going into the kidney to produce urobilin. So that's how your bilirubin is so important from various perspectives. Even in the patients with the jaundice, their feces has also been colored to the white color instead of yellow in the normal case. So this is all happening because of the bilirubin deposition. So to test this test, our this thing we to test the jaundice, there is test called Vandenberg test. So it is helpful in determining the type of bilirubin present in the serum. Normal serum gives a negative Vandenberg test, and it is performed using the diazo reagent, which is a mixture of sulfanilic acids and hydrochloric acids and sodium and nitric acids. When a diazo reagent is added to the serum containing conjugated bilirubin, a purple coloration is obtained within the 30 seconds. And this direct positive is the reason for your Van der Berg reactions. This is your Van der Berg test being explained. So, what are the causes of jaundice? What are the various etiological classifications of jaundice? It could be hemolytic, that is intracorpuscular defects because of hereditary spherocytosis, or hemoglobinopathies like sickle cell anemia, beta thalassemia, or extra corpuscular defects could be happen. Infections could be malaria, like drugs, burns, poisons, mismatched blood transfusions. All these could happen. And then hepatocellular hepatic is done in a way that its infection is viral hepatitis, malaria, typhoid, septicemia. Toxics are anesthetic agents, intratubular drugs, metals, and cirrhosis is also possible by portal and biliary. Whereas obstructive jaundice is post-hepatic, is done in the stones, strictures, parasites, see ahead of pancreas and biliary atresia. So congenital are of two types, unconjugated and conjugated. In the unconjugated ones, there is a disturbance of bilirubin transport, that is known as your Gilbert syndrome, and there is a disturbance of bilirubin conjugation happens, then it is known as Crigel-Najjar syndrome. Further, in the conjugated disturbance in the excretion of bilirubin, then it is a Dubin-Johnson, that is called as also syndrome was also known as Johnson syndrome and Rotor syndrome. So this is how threes are being differentiated nicely here in this diagram: hemolytic jaundice, hepatic jaundice, and obstructive jaundice. That is pre-haptic, before, then haptic, it's happening, then post-haptic, afterwards. So here we can see excess breakdown of RBCs of producing unconjugated bilirubin in the amount of more than healthy liver can conjugate it and excrete, and whereas in hepatic is inability of liver to efficiently conjugate and transport bilirubin into the bile due to the liver cell damage, whereas the post one is done because of the bile duct, so obstruction of the bile duct, so conjugated bilirubin can't flow through the biliary tract freely, resulting in the increased serum conjugated bilirubin. So types of serum bilirubin accumulated are unconjugated hyperbilirubinemia, whereas both unconjugated and conjugated bilirubinemia is increase in serum, and in the obstructive jaundice is a conjugated hyperbilirubinemia. So we can do the test, Van test. So it's give a indirect positive test. In the second case, it's give a biphasic reaction, and third is direct positive reaction. So urine, how is urine in bilirubin and urine urobilinogen? So these are explained in details. Please have a look. Then how is the fecal fat level, peripheral blood film? So in the fecal bilinogen, marked increased in this case. Here is a reduced, here is absent, here it's normal, here is increased, here is increased. Whereas the peripheral blood films are anemia is normal and normal. Then these are the various factors that you can look for further. Further clinical characteristics in the three diseases of jaundice, that is your preheptic and heptic and obstructive jaundice is our that is tender liver, spleen, gallbladder. Its respectively will be absent, present, non-palpable. That may be present, not palpable, absent, may be palpable. So these are the various things. Stool color will be dark brown colored stool, pale feces, hair will be clay colored stool. So different things are reason for these diseases. So breast milk jaundice. So few babies are exclusively breastfeed develop jaundice in the second week of life and may continue in the third month. So it is presumed to be due to inhibitory substances in the breast milk that interfere with the bilirubin conjugation like paragonal and free fatty acids. A bilirubin level of over 20 milligram per deciliter may be obtained, and temporary interpretation of breast milk feeding reduces the serum level of bilirubin. So pathological jaundice in newborns appears within the 24 hours of age. Increase of bilirubin is more than 5 milligram per deciliter per 24 hours, and total serum bilirubin is less than 15 milligram per deciliter, and also direct bilirubin is less than 2 milligram per deciliter. Hemolytic jaundice in newborn. This condition results from the incompatibility between maternal and fetal blood groups. Rhesus positive fetus may produce antibodies in the rhesus negative mother, and the rhesus incompatibility first child often escapes and subsequent child would be affected. Sometimes the child is born with a severe hemolytic disease, often referred to as erythroblastosis fetalis, or when blood level is more than 20 milligram per deciliter, the capacity of albumin is bound to the bilirubin, which is exceeded afterwards. So you are able to understand what is jaundice is, what are the various classification of jaundices are, and how to evaluate and treat them actually. So these were the books that has been used to generate this. So thank you very much for being part of this lecture. So let's go on to the next one. Mhm. Hypothyroidism. So students, are you all awake? All good so far? Okay, good to hear. Awesome, awesome. So now about hypothyroidism. Let's start with the hypothyroidism. Here, we'll be dealing when the thyroid is hormone is less in our body. Mainly there are two types, primary and secondary. In the primary is because of the T3 T4 level is down, and also the TSH, the thyroid stimulating hormone. In the secondary is mainly the because of the thyroid stimulating hormone has been down. So we will discuss its various etiological way and how it is done, and we will discuss more about this then. So objectives are to introduce hypothyroidism to the students. We will let the student understand what is hypothyroidism is, and to understand the cause of hypothyroidism, and to study various approaches to the treatment. So introduction is the thyroid gland regulates the metabolic function of the body in virtual every cell. Everything from the brain to skin is affected by the hormone made by the thyroid gland. Now hypothyroidism, it is a slow down it makes you lethargic and fatigue. Your hair becomes brittle and your skin becomes dry, and you become cold much easier than the average person is. So hypothyroidism is a condition where is a reduced production of thyroid hormone, categorized as primary and secondary on the basis of its cause. Primary hypothyroidism occurs due to the improper functioning of the thyroid gland, may be further classified as overt and subclinical hypothyroidism. So affects approximately 5% of individuals, with the elderly women being the most commonly affected in that case. So there's also that was a primary, and secondary one is an inadequate production of your thyroid stimulating hormone, maybe due to the congenital or acquired defects in the pituitary or hypothalamus. It is rare and occurs in less than 1% of the individuals. So primary hypothyroidism cause, what is the cause of it, etiology of it? It is because of the autoimmune thyroiditis, the Hashimoto thyroiditis it's called as, and also congenital absence of defect in the thyroid tissue. Moreover, due to the thyroid removal by the surgery, or radioablation by radioactive iodine, or destruction of thyroid tissue during the ineffective disorders like amyloidosis, sarcoidosis, various disorders are happening. Due to that, also it could happen. Uh, yeah, because of impaired synthesis of thyroid hormone, it's also possible. So, such as iodine deficiency, most common cause is congenital enzymatic defects. Further, drug mediated is the thianomides, midoarones, lithium, aminoglutathamide, and carbazamole. So, these are the drug mediated impaired synthesis of thyroid hormones could also happen in our body. Secondary hypothyroidism etiology includes the reason behind the secondary is mainly your TSH hormone, thyroid stimulating hormone. Their reduced secretion is. So there's a hypothalamic disorder such as tumor could be the reason. Inflammatory disorders such as sarcoidosis, hemochromatosis, histocytosis, and also because of hypopituitarism, that is a pituitary hormone from a pituitary glands are being produced less, leading to the mass lesions, pituitary surgery, pituitary irritations. These are the various reasons behind the hypopituitarism also. So some are following are the clinical manifestations, that is the symptoms. Tiredness, you feel very tired, weakness is there, weight gain is there with poor appetite, dry skin is there, cold sensation, hair loss, growth is retarded, constipation, poor concentration. So if all any of these things are happening to you sometimes, so might be having your hypothyroidism is there with you. Uh, so just check. Uh, I would recommend after 6 months in a year, get your all blood tests done, checked. Nothing will come for sure because you're all young guns there, bright stars for India, and I'm pretty much sure that things will be fine. But still, we should check our things from that. So, what are the clinical manifestations, signs that leads to this disease? There could be hypertension, slow relaxation of tendon reflex, edema, puffing of face, peripheral extremities. All these could be the reason because of this hypothyroidism. It could be also happen in children's, which could lead to the delay growth in the children and delay appearance of the permanent teeth. Hence, there is a precocious puberty, pseudo hypertrophy of the muscles. So. So laboratory diagnosis is in the overt hypothetism, there is a T3, T4 decreased and TSH is increased in that case. In the case of subclinical hypothetism, there is T3, T4 remain same but TSH is increased. So you can check the estimation of free T3 and T4, test for thyroid autoantibodies and thyroid scan ultrasonography, and serum cholesterol is increased in the hypothyroidism also. So treatment overview to mimic the normal psychological levels and elevate signs, symptoms and biochemical abnormalities. Treatment should be tailored to the individual needs. Treatment of choice, that is levothyroxine replacement therapy LT4 and desiccated thyroid hormone, that is T3 and T4 mixture. Insufficient evidence are not recommended for the replacement therapy by the AAC guideline. So these are the reference values for your thyroid function test TFT in pregnancy. So these are told you in the trimester, various their trimester normal value in the non-pregnant woman, FT3, FT4 and TSH, the three different readouts thyroid hormones. So in normal cases, their values are 3.7 to 7.2, 12 to 23, and 0.27 to 4.2. But in the three different trimesters, so it ranges from 1.9 to 5.18. So there's a lot of fluctuations happening. So that's why doctors recommend that to give you thyroid hormone from the outside during the pregnancy, so that there should be no much fluctuation for your hormones. So they should remain not so higher or not so low, but at the average level. So that's why most of the pregnant women or other before to keep the hormone level of thyroid to keep it good, because it might be defective, it might be not good for the kid then inside the womb of the mother. Then there's a congenital hypothyroidism that is happening in the children's. So what are the various features for that? That is, there is hypothermia, growth failure, poor feeding, uh, bradycardia, that is delayed eruption of permanent teeth, jaundice, that is muscle pseudohypertrophy, and large posterior fontanel, which is the delayed or precocious puberty, umbilical hernia leads to the pituitary enlargement. So these are all congestional hypothyroidism reason behind it. So your comorbid conditions, the hypothyroidism is quite related to your infertility, depression, hyperlipidemia, and autoimmune disorders. So outcome is that you are able to understand now about hypothyroidism, you are able to understand about the cause of hypothyroidism, and you are able to understand how you can approach for the treatment for it, various tests that we have discussed. So that was it from this perspective. Uh, I want to take you now to the, before I take you something else, to the PCR, to the virtual lab of PCR. So this is a virtual lab of polymerase chain reaction, which is a relatively simple and inexpensive tool to get the focus of segment of DNA and copy it in billions of time over. So PCR is used in every way to diagnose diseases like coronavirus also nowadays, RT-PCR being used, and also to match criminals to crime scenes and in many other ways. So this is the setup for the virtual lab bench and see how it works. Let's start. PCR virtual labs. So this is human genome made up of 3 billion chemical base pairs. Scientists have often need to isolate a very specific segment of DNA with a vast amount of genetic material. Since this segment is very tiny pieces, so we need to increase its copies to have enough copy for our work. So this is our cell nucleus. This is our DNA. So these are the various copies of your DNA, like 100 billion identical copies of DNA, that in a matter of hours, and we can use it to do a various experiments with just a few simple reagents and source of heat. So we just need simple reagents and sources of heat to do so. Not so many things, very high end things we need to do so. So let's see. So this is your hair. So to perform PCR reaction, we need a DNA that has been extracted from the cells. Because the purpose is to make DNA copies, you don't need very much DNA to start the reaction. So we could extract DNA from small sample of blood, skin, saliva, or even hair follicles. So let's start by moving your extracted DNA into special PCR tubes. PCR works by heating and cooling solutions over the PCRs so that we can start our reaction. So to start PCR, let's drag the extracted DNA from yesterday. We did this isolation of DNA into the PCR tube. Let's take this and we put them into the PCR tube. So now let's add primer 1 to the PCR tube. So primer 1 will attach to the DNA strands that are at either end of segment, and that is the powerful tool for copying very specific DNA. So let's add that. So we take the primer 1 and into the PCR tube. Now add primer 2 to the second side of your DNA. So now all the primers are being added. Now it's time to add the nucleotides, that is your A, C, G and T, to make up your DNA code. So we add this nucleotide of the PCR tube, the nucleotide PCR tube here. So last we will add our DNA polymerase, which will help us to make many copies of our DNA that we want to run. DNA polymerase. So now the PCR tube is filled with all the reaction component. You will place it into DNA thermal cycler, and this machine will precisely heat and cool and your tube at specific times during the next hours. These changes in temperature are crucial for making the reaction works. So drag the PCR tube into the thermal cycler. So here temperature will be taken care for you. Now let's start. So this is your target sequence. So we make the temperature of 95 degrees Celsius first inside the thermocycler, and it will unwind our target sequence. And this is your primer 1 and primer 2 being attached here, and the temperature is made 50 degrees Celsius here. So and they get locked in that situation. Then we take the temperature to 72 degrees Celsius so that DNA polymerase could be attached and could run the reaction. So click, this is was the cycle 1. The same way we do the temperature again down to 50 degrees Celsius. Oh sorry, 95, my apology. 95, then 50, then 72. Then again your action is done. Then we click on next. So after 3 cycles, this will be your desired fragments first being used there. So at the end of a cycle 4, you will have 8 fragments that contain your target sequence. So now we have 22 fragments that contain only your target sequence and only 10 longer length copies. Click this. So you see how many copies of DNA has been arranged with 30 cycles. So after 30 cycles, there are billion of fragments of the same copy of DNA that only 60 copies of longer length molecules. So you now have solution to nearly pure target sequence. So you have whatever you want to work for, you can work with this DNA for your further reaction. So okay, students, we had done isolation of DNA from the virtual lab, and we had done the PCR reaction to increase your DNA that you have isolated. Now it's your time to check how you can check it's how much it is being produced. For that, scientists have found a technique called gel electrophoresis. So here in this this is called as Appendorf. In this plastic tube, there is some clear liquid in it that is your DNA of several length that we have extracted with the PCR. Now our job is to check how much length is our DNA is, how much length we have produced. So we use this technique called gel electrophoresis for that. So this is your short, medium, and long, right? DNA strands, short DNA strands, medium DNA strands, and long DNA strands. So we want to check them by using a Western blot, sorry, gel electrophoresis technique. That how much production is there, how much is present. So to do so, we do electrophoresis, gel electrophoresis. So DNA strands are molecules so tiny that you don't see them even under microscopes. Is there any way to sort them so we can check them via gel electrophoresis? So scientists are using this technique to check the length of the DNA molecule. And so let's see how does this technique work actually. So basic principle, but first we will see the basic principle behind it, the basic biochemistry actually. So gel is the filter that sort the DNA strand. It is a sponge made of jello with many small holes in it. So you press forward to continue. So we place DNA samples into holes at one end of the gel, negative at and one end, positive at another end. So is your DNA is a negatively charged because of the phosphate group, it will move from negative to positive side, right? So you can see that different DNA material has short, middle, and longer. Each sample has different level of working out. So we put this gel into the UV light and we can see each strands of DNA gel in different panel. Have you done any one of you these gel electrophoresis so far? Have you done any of these experiments in your bachelor's or in master's? Did you had any experience of that, students, before? Okay, that's good. So this is very nice way to check it, how you can do it. So now let's start with making the gel first. If somebody has not done it, so this will tell you each steps very easy manner. So you can do it on your own then later. So this is your agarose gel and this is your flask we need, gel mold, gel comb, buffer and microwave. So you put small amount of agarose into your conical flask. Then put this liquid buffer to the flask so that electrical charges could flow. And it goes for microwave. We will boil it to make it into a liquid form. Now we remove the plastic wrap and put this into the our gel chamber to get it solidify. Before it's get solidified, we need to make some combs. We need to make some wells. So we use this comb on the one end of it and just add it there. So this will create some holes to put our DNA. Let it cool down. Once it's cooled down, we remove this out to set up the gel apparatus now. So in the gel apparatus, we add this mixture, buffer. So place the gel inside the buffer. So this is loading buffer, DNA sample, DNA size standard, micropipette, gel box with buffer and gel, pipette tips. So let's start now. What we do is we have a standard DNA standard size to check like how many sizes are there. This will give a standard size for running the buffer. And in the DNA sample, we will add load would be heavy and it could be visible because of the color in it. So we add loading buffer into the DNA sample. So like this, easy peasy. Now take this material again and put into your gel. So to the first column. So do this very carefully so that it should not come outside or go into the second well. These steps has to be taken care. Then you take your DNA standard size standard and you put it in here. So it's time to put onto the electricity now. So black with the black, red with the red, then turn it on. So this is the bubbles will starts to move up. So that means the your gel is running. So like this, bubble starts, your gel is running then. Then your gel starts to go away, your DNA will start moving forward. So you have finished your gel. So you take this gel and put them into the ethidium bromide solution. Okay. So that the ethidium bromide will attach to the base pairs of your DNA. So it's about take half an hour to stain the gel, and we will speed up the clock. So remove the gel and place it on the UV light and turn it on. So this is what you see, various DNA size base pairs is. That is, this one would be around 1500, and this is around 3500 base pairs, and this is around 6000 base pairs of DNA. So this is how you can calculate according to the standard. So all estimations are correct. So we are able to understand how DNA is run. However, with the help of electrophoresis, first we isolate the DNA. Then after isolation, we did the PCR. Then after the PCR, we run in sample. And this is what you see, the three different size pairs of your DNA, short, medium, and long ones. Running it like that way. So I would like to stop it here. We are done for today's lectures. I will share later the presentations of today. And yeah, I hope you had enjoyed this workshop, 2-day workshop, short and short. And I have given you some knowledge. Please take it with you and make some assignment if possible, in a good way, so that it could be beneficial for you also for the later part. Yeah. So any questions, bachcho? Any questions? Any doubts? My welcome, my pleasure. I hope the voice, the videos, the presentations, everyone think perfect well, and you're able to understand things all in all. And the theme was also fine with you, with the basics of biochemistry to the advanced version of biochemistry. So, Bache, we also gonna start with the IITs direct workshop in the area at the IITs live, but due to COVID region, everything is going online. So basically, we are at the moment working online workshops. Yes, but if you're interested, um, so there are some workshops coming up in future. So so I will be taking care of most of the workshops, but some so like there is a MBBT workshop. So this was 2 days workshop for you, but if you're interested, so it will be from 7 October to 16 October. We'll be dealing with that workshop. Then we will be dealing with the proteomic workshop, biochemistry workshop, bioinformatics workshop also. Just have a look. So this was our biochemistry, this one for today. Also nowadays my workshop is going with the plant biotechnology at the moment. So I will have a class just now after this at 7:30. So these are the various workshops that we are dealing at the moment. So after this series, I will start with the principle of genetics. If somebody is in with the genetics, please come and register for the same. So we have many workshops going on at the moment. So I can share these in the platform also later with you students, so that you can have a look if you want to continue with these things. Yeah, I will be happy to join offline also, because it's been ages that we have seen students, and students have seen professors in front of each other. But this is how the new life is. We have to adapt it, and it somehow it's working also good. You are attending so many webinars, so many workshops in a few days, so you are getting a lot of certificates. I have seen on LinkedIn, and many students are working very hard during these days also. So but this was not possible during offline time. So so you will get your certificate soon, I guess, maybe tomorrow, day after tomorrow. Okay, great, great, great. Thank you very much. Yes, yes, you will all get certificates for these workshops by Vatsal sir and Shwani ma'am. So, uh, feedback link. So I will ask Shivani ma'am to create a feedback link for this also. I will let you know about that too. So I had a really good time with the students. It was quite small days, but we did a lot, and I hope you like it. And I wish you all good luck, all the best for your career. Wherever your heart goes, you should follow that passion and take it to the brightest stars of the sky. That's my wishes for all of each one of you, and you make your career to the top notch of your life. Uh, and that's my all best wishes.